Thank you for attending this talk. I'm going to try to take you through some of our research on understanding human genetic diversity across global populations. I'm an associate professor at University of Birmingham. My group now is in two places. We are still in the main campus in Edgbaston in Birmingham, but in the past 2 years we've been moving to Dubai because we have a new campus in Dubai now, and we're also running the health data science program there. So my presentation is I'm gonna give you some background about what we do and try to tell you why it's important to study diverse populations. And the last part of my talk will be specifically on Middle Eastern populations. So the Human Genome Diversity Group at Birmingham is interested in studying diverse populations to understand human genetic diversity. And by human genetic diversity, this includes, of course, ancestry and genetic trace and diseases. So we understand genetic diversity as a product of events that have occurred in the past. So events like demographic and biological processes that have shaped over time the genetic diversity that we observe today in different populations. The whole talk, I'm going to speak about data, which is going to be whole genome sequencing data, but we also use non-genetic data, which is clinical reports, historical data, linguistic data, archaeology, anthropology, and of course we wrap up everything using bioinformatics. So whole genome sequencing for us population geneticists has been a game changer because of how much information you can get from whole genome sequencing. So now we know from sequencing diverse populations that we have all started, so all modern humans have started as one population in Africa around 200, 300,000 years ago. And we also know from sequencing that around 60,000 years ago, people exited Africa and went to occupy the whole world. Now, what is very, very interesting is that each one of us today, we still have genetic signals of those past events, even if they have occurred thousands of years ago. One of this information, for example, is that we can know from a single genome what was the population size thousands of years ago. So in a plot like this one here, we have on the x-axis the time, years ago. This is like 10,000 years ago, and this is like 100,000 years ago. And we can see the population size here. And every line here represents a genome from an individual that comes from a different population. A feature that you can see in this plot here is that not all non-African populations have this bottleneck around 6,000 years ago. So a bottleneck, which is a decrease in population size around 60,000 years ago. So just few thousand individuals exited Africa, taking with them is just a subset of the African genetic diversity. So everyone outside Africa is a subset of the African genetic diversity. And to give you a perspective of how much less diversity we have outside Africa than in Africa, when we looked at population in Chad, we studied different ethnic groups there. And some of them, like the Sara and Lal, they are typical sub-Saharan African populations. On the x-axis here, we have the Eurasian ancestry, and those populations would have like 0% of Eurasian ancestry. They are typical sub-Saharan African populations. And we're measuring here heterozygosity. So heterozygosity for us is a measure of genetic diversity. Probably you all know what heterozygosity is. It's a specific part of the genome. The maternal and paternal allele are different. So more differences means a more diverse population. So you can see here population like sub-Saharan population, Sara and Lal, much more diverse than Eurasian populations like Lebanese and Greek. What was very, very surprising to us is finding populations like the Tubu, their genome is a mixture now of African and Eurasian ancestry. So they have around 30, 40% of Eurasian ancestry from a migration that happened 6,000 years ago to Africa. This is very surprising to us because if you look at population genetic rule, when you mix two populations, the resulting population should be more diverse than the two sources. So this Tubu should be here, somewhere here, but they are intermediate. And when we looked at the Tubu genome in more details, we found now, over time, their genome is a mosaic of different segments. So over time, the combination broke their segments into what looks like this now. So we have African segments who have the typical genetic diversity of African, sub-Saharan Africans. They have the African Eurasian segments who actually have more diversity than what you expect from African Eurasian alone. So this is what would place Tubu here. However, they have also Eurasian segments now, and those are so much depleted in genetic diversity that the overall result is placing the tubo as intermediate population. This is just to show you how much less genetic diversity we have outside Africa than in Africa. But humans, even with this like small genetic diversity, they exited Africa and they went and occupied the whole world, adapting and living at different environments. And one of the most harsh environments that we've studied is living at high altitude. So living at high altitude is a very, very taxing phenotype. If you go to 3,000 meters, you're probably going to have a hard time doing regular exercises. Going to 5,000 meters, you're going to have some memory loss. A little bit higher, probably you're going to lose consciousness. However, you have populations in the Himalaya who were born there, who live there, and they spend all their life there. So they have adapted to live at high altitude. And of course, when we looked at their genomes, we found that they have a lot of mutations in genes that allow them to live at high altitude. So this graph here, those are human chromosomes here. Every point it represents a SNP, and the more differentiated, the higher it is, it means it's more differentiated in high altitude populations. So this shows you like you have a lot of genes that were modified to allow Himalayans to live at high altitude. So this is not surprising. However, it's very, very interesting when we compile all this data, this information, with also what other people were doing, and also look at studies that were coming from Ethiopia for people who lived at high altitude. People found that Ethiopians also have genes that were modified to allow them to live at high altitude. Some of those genes that were modified are shared with the Himalayans, some of them private to the Ethiopians. And also people were studying people who lived at high altitude in the Andes. And those also had modified genes. Some of them shared with the Himalaya, some of them with Ethiopians, some of them with tree populations, but some of them private to the Andes population. So this shows you that we have one human phenotype, one human trait, which is living at high altitude, but 3 different populations have reached this phenotype, modifying different genes through different mutations, different variation on their genome. And this is not an exception in human evolution, an exception in how people have developed their diseases or traits. An example that I like to give is lactase persistence. So us as humans, as adults, we cannot digest milk. However, there's a mutation in a gene called MCM6 that allows people to digest milk. And this mutation frequency in Europe is almost 100%. So if you are European, definitely you have this mutation that allows you to digest milk. And the frequency of this mutation outside Europe is 0%. So this is why for a long period of time, drinking milk was associated with white supremacy. So if you go on YouTube now and Google drinking milk white supremacy conventions, you're going to see people drinking milk in those conventions. So, however, when we studied East African and Arabian populations, we found another mutation in the same gene, MCM6 also, that allowed people there to digest milk. And the frequency of this mutation is 0% elsewhere. So drinking milk is not exclusive to one population. So from the 2 examples that I showed you, you probably now understand that if we want to study any human traits or any diseases, we should be studying diverse populations, all human populations, basically. However, unfortunately, this is not what we were doing. So those plots here, they show you genome-wide association studies. So those are studies where they try to link a mutation on genome, a variation on genome, to a certain trait and diseases. And you can see that the participants for those studies, they come just from one population. So we were not studying diverse population, we were just studying one population. And this puts a lot of limits about how we understand human diseases. So this was honestly one of the main incentives for us to study Middle Easterners, because the Middle East is one of the places that is the most underrepresented in genetic studies. And in a series of papers starting from 2017 until recently, even a little bit before 2017, we tried to explain how populations in the Middle East have formed, including their ancestries and some of their diseases. If you remember at the beginning of the talk, I mentioned whole genome sequencing as this amazing tool that allowed us to get a lot of information. Now there's another also very powerful tool that we're using, which is ancient DNA. So ancient DNA for us was also a game changer, because with ancient DNA, before ancient DNA actually, when we looked at genetic diversity that we have today, we were trying to put models, we were trying to predict what happened in the past and for us to see what we have today. However, with ancient DNA, we can simply go and sequence the populations that lived and died thousands of years ago. So we can look at how the genetic changes were occurring over time, and we can understand why we have them the way they are, they are today. So with this amazing tool, we constructed what we call the genetic timeline of the Middle East. So in this timeline, we try to explain how modern populations here, you can see it at the bottom of this graph, modern populations like Lebanese and Emiratis have formed over the time. And this graph here, it shows you all their ancestors and how those ancestors were related and how they descended until they became the Middle Easterners today. So this graph is built, you don't have to know the names on this graph, but this graph is populations, ancient populations that we and others have sequenced. And I'm gonna guide you through this graph. So as I mentioned before, Middle Easterners and all other human populations, they coalesced around 200, 300,000 years ago. And Middle Easterners are not an exception to this. At 60,000 years ago, also I mentioned, people exited Africa. And when people exited Africa, they met another human species called the Neanderthals. So today, all modern humans who are living outside Africa, we have around 2% of our genomes coming from Neanderthal. So modern humans and Neanderthals, they have a common ancestor around 500, 700,000 years ago. And when people exit Africa, they mix with Neanderthals, and we all have, if you are non-African, you have 2% of your genome coming from Neanderthals. This is not our study, by the way. I'm citing this study from 2010. However, what we found is that Middle Easterners also have this Neanderthal ancestry. However, Middle Easterners have the least amount of Neanderthal ancestry among all human populations. And the reason for this is we found out that as soon as people exited Africa, this human population quickly split into two populations. One is the main Eurasian ancestry, the main Eurasian population that mixed with Neanderthals and then went and occupied the whole world. But then there was a basal Eurasian ancestry that stayed in the Middle East, did not mix with Neanderthals. And the ancestry of modern populations today still derive from this basal Eurasian ancestry, some of it. It's a mixture of both main and basal Eurasian ancestry. So it's amazing to see that modern populations today still derive from this like very early population that exited Africa 50, 60,000 years ago. So around 40, 20,000 years ago, there was an ice age. And we can see also from genetics that populations in the northern part of the Middle East and the southern part of the Middle East, they were locked into two different populations now. And around 10,000 years ago, there was an invention in this part of the world that basically changed everything, the way we do stuff in terms of our societies and in terms of our culture. This invention was invention of agriculture. So before farming, before agriculture, people lived in hunter-gatherers. As hunter-gatherers, they lived in small bands of 100 people going from place to place, moving from place to place. However, with the invention of agriculture, people had to settle. And to maintain agricultural societies, you need a large number of people to maintain it. What's amazing here is that we can see this expansion in human populations and from genetics. So populations who live in this part of the region here, Syrians, Lebanese, and Iraqis, you can see from their genetics an expansion in the population around 10,000 years ago. What's also interesting is that Arabian populations like Emiratis and Saudis, we don't see this expansion. And this is because at this time, those populations did not move to farming, so they did not start farming, but what they did was start camel herding. And if you remember the mutation that I mentioned before, the MC6 mutation, so this mutation started increasing around this time. So we can detect the increase in frequency of this mutation through time, and this mutation started increasing at this time because we think people started drinking camel milk. So in Europe, probably people were drinking goat milk. Here in Arabia, people were drinking camel milk, and this mutation was very beneficial for the population, so it increased in frequency. Around 6,000 years ago, we see another bottleneck in the Arabian population. So just the Arabian populations had this bottleneck around 6,000 years ago. And this is because around 6,000 years ago, this place here became a desert. So before 5,000, 6,000 years ago, this place was a green landscape. And desertification started around this time. And we can also see it in the genome of people who are living today. So around 5,000 years ago, This was the time when Semitic languages, so Semitic languages like Arabic, Hebrew, those languages started spreading in the Middle East. And linguists have also a methods to detect how the languages are spreading from place to place, like us geneticists. And when we put the information from linguistics over the information from genetics, we can see that there's almost complete overlap. So now we know that languages, Semitic languages like Arabic, spread by people moving, not just by ideas. So it was people moving who spread those languages in Arabia. So around 4,000 years ago, now populations have established. And if you look at the genome of someone who lived in Lebanon around 4,000 years ago and someone who is living today, their genome in terms of ancestry is almost identical. There's very little change 4,000 years ago until today. However, it's not only ancestry. So even if we look at the frequency of certain variants that are related to certain diseases, we can also see that the frequency of those mutations are very similar 4,000 years ago and in modern populations. So people who lived 4,000 years ago, they almost shared the same disease mutations that people living today have them. And this is a little bit surprising for us because you would expect disease variants to decrease in frequency over time. But this is not what we're seeing here. Actually, we're seeing something opposite. So in the Emirati population, we see that variants, so hundreds of variants that are related to type 2 diabetes were actually increasing. So over 2,000 years, they were increasing in frequency in the population. And you probably know that the Emirati population has one of the highest prevalence of type 2 diabetes in the world. And it was always thought that this is caused by a change in lifestyle and change in diet. However, what we showed is actually there's some genetic background that probably was beneficial at one point. But now it's causing type 2 diabetes in modern populations. So another mutation that spread around this time is a mutation that actually is protective to malaria, and it spread specifically in Eastern Arabians, so Emiratis and Omanis. And this mutation is protective to malaria, and it spread, the increase of this mutation is around 5,000 years ago. However, by 2,000 years ago, so when we sequenced ancient Bahraini who lived 2,000 years ago, everyone had this mutation. So it shows you how important this mutation was. And when we looked at when agriculture also started in Arabia, so agriculture in Arabia started very late. So it started around this time. So this was probably very, very protective for those populations at this time. And in the last 1,000 years, so after those populations have formed, now what we started seeing is population structure, so differentiation inside the populations themselves. And this is a PCA plot, so probably also you know what's PCA plot, but every point here represents an individual. And the difference between those two points or two individuals is basically related to their genetic relatedness. So if they are closer on the plot to each other, it means they are genetically closer to each other. If they are far, it means they're genetically distant to each other. So this is a plot, this is a PCA from Lebanon. And in Lebanon, what we see population structure is being caused by religion. It's almost perfect explanation of the genetic structure in this area. And this is because we have non-random mating, right? So we have a preference in one group to marry from the same group. So this creates population structure. So this is in the northern part of the Middle East. In the southern part of the Middle East, we have one religion, but the population structure seems to be related to tribal affiliation. So this is very similar to PCA here, but those are branches. So also if two branches are close to each other, it means that they're genetically related. So you can see here, those are the regions, but inside the regions, you have the tribe. So affiliation or population structure in the Emirates is based on tribal affiliation. Then those tribes themselves are associated with certain geographies and certain places. And basically the overall structure in the Emirates is tribal affiliation and tribal structure for this population. And this information is very important if you're going to do any medical studies. You need to understand what is driving population structure in this population. So from this talk, I just want to take 2 points here, which is we as humans, we all derive from same population, from one population. However, we have differentiated, and there are some small differences between populations who are living today, but those small differences are sometimes medically relevant. And if you want to understand any human trait or disease, you need to study, and gene function, of course, you need to study diverse population, not just one population. So thank you for listening.